Good afternoon, everyone. Today's topic is selection of recombinants. In biotechnology, one of the commonly used vector is PBR322. It is an artificial vector which contains two important sites that will help us in selection of recombinants. What are those important sites? Ampicillin resistance gene and a tetracycline resistance gene. What is ampicillin? Ampicillin is an antibiotic, which means an antibiotic is commonly used to kill bacteria. You have ampicillin and tetracycline, which are the antibiotics which are used to kill bacteria. But speciality of PBR322 is it carries resistant genes against ampicillin, which ampicillin and tetracycline, which means say you are having a bacteria which contains PBR322 in it. That means you cannot kill that particular bacteria by pouring ampicillin as well as tetracycline antibiotic because of the presence of resistance gene. In this tetracycline resistance gene, upon it, it is having one restriction site or a cloning site. What is restriction site or a cloning site? It is a site where you can make cut by adding particular restriction enzyme. Look at here, I have shown one site known as BAM-H1 site. BAM-H1 is a restriction enzyme like EcoR1. If you add BAM-H1, it will make cut in the entire PBR only here in the tetracycline gene, which means when you cut the tetracycline gene at the BAM-H1 site, tetracycline resistance is lost. That is the important feature. You have to remember about PBR322. Now let us go into the experiment, how to select the recombinants, what is meant by recombinants, transformed cell, non-transformed cell. Let us understand. In a test tube, take PBR322 vector which is required for our recombinant experiment. To that, add restriction enzyme BAM-H1. You know BAM-H1 will always make cut upon the tetracycline gene at this particular site. So a nick is made in the tetracycline gene. Here you can observe that after adding BAM-H1, cut is being generated in the PBR. Now to that PBR which is having cut, add your desired gene or gene of interest. You can call it as foreign DNA. You can take example of, let us say, insulin gene. Take insulin gene as a foreign gene, add it. Since cut has generated sticky ends, there is going to be combining of foreign gene to the site where you have made cut. But because of cut in the plasmid, there can be possibility of self ligation. Instead of your foreign gene sticking onto that place, the PBR itself would have self ligated. As a result, in this test tube, upon adding foreign gene, you will be having two types of plasmids. One which is not having any foreign gene in it, it is known as non-recombinant. It has not combined with any of the foreign DNA. Second type of plasmid you are having is plasmid with the foreign DNA. Look at here, red colored one is a foreign DNA and plasmid is intact here. So two types of non-recombinant and recombinant plasmids we have got. Next step is, once you in your test tube, now non-recombinants are there, recombinants are there. Now transfer it into the bacterial cell like E. coli. E. coli is commonly called host cell. How to transfer? Use heat shock method. By using heat shock method, when you transfer these restriction and endonuclease treated plasmid randomly into the bacterial cell, you will end up getting three kinds of cell. What are those three kinds of cell? First type of E. coli cell, which might not have taken any plasmid inside. That means it has not transformed itself, hence such E. coli cell without any plasmid is known as non-transformed cell. Second type of E. coli cell, it has taken the plasmid but non-recombinant plasmid it has taken, hence it is a transformed bacteria because it has taken something transformed but with non-recombinant plasmid. Third type of E. coli cell is the one which has taken the plasmid which is recombinant, having foreign gene. Here it is shown. Hence, that particular bacteria is known as transformed bacteria with recombinant DNA. Now, out of these three, which one we require for our experiment to grow further, to get the product, to get the desired product? That is, we don't need non-transformed cell because it does not have foreign gene. Transformed cell with non-recombinant also we don't want because there is no foreign gene. We need third kind of cell, transformed cell with the recombinant DNA. Hence, we have to eliminate non-transformed cell, transformed cell with non-recombinant DNA. We have to eliminate and select only transformed cell with recombinant DNA. That is known as screening, selecting only required cell. Screening is done in two ways: replica plating technique and blue-white colony. Replica plating technique: take a Petri plate, take normal medium, grow all these cells which are present in the test tube. Now you do not know which cell is having recombinant DNA, which is not having, you do not know. So grow them in a normal media. Normal media, when you grow non-transformed cell, I have written it as NT, non-transformed cell, black colored colony, they will grow, produce their colony. Transformed cell with recombinant, with non-recombinant DNA, they will also grow. Transformed cell with the recombinant DNA, they will also grow. So all three kinds of cells will be growing in a normal medium because there is no antibiotic to kill these colonies. Now take a Petri plate. In that Petri plate, or to the same Petri plate also, you can add ampicillin medium. When you add ampicillin medium, now what will happen to non-transformed cell? Non-transformed cell, it doesn't have plasmid inside, which means it doesn't have either ampicillin or tetracycline. Upon addition of ampicillin, since there is no resistance, non-transformed colonies, the black colored colonies, will not appear in the Petri plate. It is now eliminated completely. Transformed cell with non-recombinant plasmid. Transformed cell means it is having plasmid, having ampicillin resistance gene. When you add ampicillin, because of the resistance, it will grow in the medium. Third one, transformed cell with recombinant DNA. Recombinant DNA means you are cutting only the BAM-H1 tetracycline gene. Tetracycline resistance has been lost, but ampicillin you have not touched at all. It is intact, hence they will be having resistance. Therefore, they will also grow in a medium containing ampicillin. Now an important method whereby it is as the name indicates, method is replica plating technique. Create a copy of the second Petri plate, which is where you have poured ampicillin. How to create a copy? You consider that you are having one Petri plate, take another Petri plate, copy it, colonies are copied. Actually, it is not done like that. To simplify, I am giving an example. Okay, you are taking one Petri plate, copy it. When you copy it, all colonies will be copied, but now to the copied replica plate, this is called replica plate. Now to that you pour tetracycline medium. When you pour tetracycline medium, anyhow non-transformants have colonies have been eliminated in second case only. Now transformed E. coli with non-recombinant DNA, they will grow in tetracycline medium in the copy plate. Why they will grow? Because these have intact plasmid, their tetracycline gene have not been cut at all. Hence, they will grow. Hence, blue color indicates the transformed cell with non-recombinant DNA will be growing. Wherever, look at the colony. In the second plate, where they are growing, same colony. One colony, third colony, 18th colony, then 7th colony. So same place they will be growing because we have copied it. But whatever red colored colonies are there, red colored is transformed cell with recombinant DNA, they will disappear now. Why do they disappear? Because you have added tetracycline to the Petri plate. Tetracycline resistance is seen in the transformed cell with the non-recombinant DNA, whereas transformed cell with recombinant DNA, we have made cut in the BAM-H1 site of tetracycline. As a result, resistance is being lost. Since resistance is not there in the presence of tetracycline, they will not survive, they will die. But you may think, for our experiment, I said in the beginning, we need only transformed cell with rDNA. How to take it? Now you know the blue colored colonies here in my diagram, that is transformed cell with non-recombinant DNA. Now trace back. Tracing back means go to the previous plate. In the previous plate, wherever colonies are there in the same place, corresponding to same place, belongs to the, it indicates that these are non-recombinant cells. Whereas wherever colonies are disappeared, it means that these are the colonies containing foreign gene in it. They are the one which is required for our experiment. So let us take the example. Say this is the 10th colony. I have marked it as 10. Here 10 is missing. So since when you observe, you will find that 10 is missing here. Hence you can trace back, get this 10th colony. Take this colony out, grow them in a bioreactor, you will get the desired product like that of insulin gene. So this is how replica plating technique is done. Second method is blue-white colony. Compared to replica plating, blue-white colony is the easiest method. Take a vector like PUC18. PUC18 has ampicillin resistance site and a gene known as LAC-Z gene. LAC-Z gene codes for an enzyme known as beta-galactosidase enzyme. Upon this LAC-Z gene, there is a cloning site or a restriction site where you can make cut and you can insert your foreign DNA. Similar to tetracycline resistance, upon that BAM-H1 was there. Now LAC-Z gene is having cloning site where you can insert your foreign DNA. Now in the experiment, take this PUC18 vector, make cut in the cloning site by treating them with particular restriction enzyme, insert your foreign gene in it. When you insert foreign gene in it, you do not know whether foreign gene has linked to the LAC-Z site or self-ligation might have taken place. You do not know. So you have to find out recombinants from that of non-recombinants. How do you find out? It is easy method. It is based on the chromogenic substrate. What is chromogenic substrate? Chroma means color. Grow them in a particular medium. Based on the color, you identify transformed cell from that of non-transformed cell. Which is that chromogenic substrate? It is known as X-gal medium. In the X-gal medium, grow the cell. If bacterial cell, if the bacterial cell is having, and it is not, if it is not having any insert, which means LAC-Z gene is intact there, it will produce enzyme. So enzyme will react with X-gal. X-gal gets converted into blue colored colonies. Look at here, I have shown PUC18, no insert at all, no foreign gene, hence you will get blue colored colonies. Next, if the plasmid is having insert, look at here, I have shown one insert, then while growing on X-gal medium, because of insertion of foreign gene, you have inactivated LAC-Z gene. This is called insertional inactivation. What is insertional inactivation? By inserting the foreign DNA in the LAC-Z gene, we are inactivating the LAC-Z gene. When LAC-Z gene is inactivated due to insertion, beta-galactosidase will not be produced. Hence, X-gal, in the absence of enzyme, it will produce white colored colonies. Look at here, white colored colonies. Now, in the Petri plate, there are two colonies are present. That is blue colored colony and a white colored colony. what I need is only white colored colony, because I know white colored colony means these cells will be having insert. Take out that, grow them in a bioreactor, you will get the desired product. So these are the methods of selection of recombinants. Thank you. If you have any doubt, please mail me.